jet that comes next week with our B lymphocytes. I believe it's on Thursday, Tuesday or Thursday. But your body cells have attached to them a marker, usually proteins, but they can have glycogen or lipid attached to them. All right. Typically, these markers are unique to your body, and that's why your body would develop antibodies against them if they were foreign. So that's why with a heart transplant or a kidney transplant, you have to take immunosuppressive drugs to suppress that development of the lymphocyte reaction against those foreign antigens. Our red blood cells have marker antigens on them as well, but these antigens are shared among different individuals. There are smaller, less populous, less frequent antigens that are present that you'll learn about in micro. But the three antigens we're going to look at today are the typical blood typing ones: antigen A, antigen B, and the Rh factor currently known as antigen D, as in doc. There's E and F and C and other ones, but we're not going to look at those. So an antigen is kind of a circular reasoning definition. An antigen is identified as a molecule which triggers an immune response. An immune response is the production of antibodies against an antigen or another chemical release, or it's a response the body gives to an antigen. So it doesn't really tell you a whole lot. But an antigen can have different surfaces, and usually an antibody will recognize one or more of those surfaces. An antibody is a protein that's shaped like a Y. We'll look at its structure next week. It actually has two chains, one long chain and then two short chains, but it still looks like four chains, but it looks like a Y. And so if we have an antibody, it has unique amino acid ends, and those recognize and are specific to the sequence of amino acids on the antigen. So we have this antigen-antibody reaction. The antibodies are made by B lymphocytes. When you are born, you do not have antibodies in your blood, but many of the foods that we eat have the antigens A and B on them. And so within the first 6 months of your life, you will develop antibodies against A if you have antigen B. You don't develop antibodies against your own antigens. So if you have antigen A, all right, your body will develop antibodies against the antigen you don't have. So you will develop antibodies against B. So your bud, after about the age of 5 to 6 months as a baby, will have B antibodies in it. Again, developed from the food against the bacteria or the food products that you eat. And we'll talk about this. These antibodies that develop against this are of a large, they're actually clustered together in clumps, so they're quite large, and they won't cross the placenta usually. So we'll talk about that in a few moments when we talk about erythroblastosis totalis. If you have antibody B, then your body will have the antibodies. It will develop by the time you're about 6 months old. It will develop the antibodies against the antigen that you don't have, which is antibody A. The third type of antigen is antigen B. Now, if you don't have the D antigen, as these individual cells that brought up here do not, if you do not have the D antigen, you do not develop antibodies against it because it's not found in plants. It's found only in primates, so even if you eat beef or fish, you're not going to be ingesting that antigen and introducing it to your body. This is the theory of where these antibodies come from. So to develop the antibody D, you would have to be exposed to blood cells that have the D antigen on them. Okay? So we're going to look at how the process occurs. So we're going to make a chart similar to what you have in your textbook. So A, people with red blood cells that have A would react to their food and make antibodies against B, but not against themselves. People that have blood cells with D antigen would make antibodies against A. And people that don't have, so neither of these have D, and they wouldn't make antibodies against it unless they'd had a transfusion with the D antigen. We describe the D antigen as plus or negative. So you've heard I'm AB positive. That means you have the A antigen, the B antigen, and the D antigen. Okay? So if you were AB positive, would you ever make the D antibody? No, because you would have the D antigen. If you were AB negative, you could make the D antibody. If you had a baby, you were a woman,
and you got a blood transfusion,
who was, had the D antigen, or if you were given a transfusion. And we'll look at specific cases and give examples of that. All right? So this next image I'll talk about just a little bit, and then I'll pull the screen up and we'll talk about diagrams. So this is just showing the A antigen and the B antibodies being present, the B antigen and the A antibodies being present. If your blood cell type has both antigens, then you don't make either of the antibodies, because you don't make antibodies against yourself. Right? If your blood type has no antigen on it, then you have both antibodies in your blood by the time you're 5 to 6 months old. Okay? Yes?
So then D has no antibodies.
Only if you are negative or positive, you would have no antibodies, unless you have that exposure. Later on,
Right.
you would get it. So let me turn off the overhead and let's work through the table. It's on the PowerPoint and it should be also in your lecture notes.
so the exposure was fetus to the mother or transfusion.
So this is not complicated, it's merely confusing with all the A's and O's and B's. So I want to go through slowly with you, and we'll work with this concept when we do this particular lab. And help to clarify the rules to follow, because there's a logic behind those rules. So blood type is determined by the antigens that are present on the red blood cell. So once we identify the antigen, we have identified the blood type. Okay? So if our blood type is A, we have the A antigen. And I'm going to work with the ABO blood type and then come back and add the D antigen concept. Okay? If our blood type is B, we have the B antigen present on the red blood cells. If our blood type is A, B, then we have both antigen A and antigen B. And if our blood type is O, we have no N.
Universal donor.
Pardon?
That's me. Oh.
O. About 40% of the population is O. Pardon?
Universal donor.
Yeah. Well, not just O, but you're close. Now, let's put up the antibodies. Also on your sheet, they're called agglutinins. So agglutinin and an antibody mean the same thing.
Oh, she's doing this.
So the antibody
The opposite of antigen.
is going to be the opposite. All right, so if you have the antigen A, you're going to have antibody against B. If you have the antigen B, you're going to have the antibodies against A. If you have both antigen and B, you have no antibodies. If you did, you'd be agglutinating or clumping your own blood cells. If you have blood type O, you have both. A antibodies and B antibodies. Again, by saying a B antibody, I'm talking about an antibody protein whose ends of the Y are going to match and bind to the B antigen. Okay? When that happens, if the B antigens are on two different cells, those cells are clumped together. And that is not clotting. Clotting is the formation of fibrin proteins that trap water molecules. This is agglutination. This clumping damages the red blood cells. It blocks blood vessels. It can cause the red blood cells to rupture. This causes a high fever, and it can result in death. It's known as a transfusion reaction. So you never want to give or accept the wrong blood type. So the next list we're going to look at, then, I'm going to switch columns. I'm going to do receive from first, because I think it's easier to understand in that sequence. So if my blood type is A, who can I receive blood from as a transfusion?
A and O.
So let's look at the concept, the reasoning, because I don't want you memorizing this table, because then you can forget it. If you understand the concept and the reason behind it, you can always figure it out. So this is your blood type here, all right? You're the one receiving the blood. And we're giving the blood. Now, the key to this is you don't ever want to have the recipient blood with antibodies against the blood coming in. Let me illustrate. If I have antibodies against B, and I add blood with B antigen, all of those blood cells are going to get agglutinated. Okay? So if these are my B antibodies, and I add B antigen, it's not going to work. I'm going to have a transfusion reaction. So you never want to receive blood that you have antibodies against. You never want to give blood to somebody that has antibodies against you. The key is the antibodies that are in the larger volume. So the person's body, the antibodies that are in the person's body, not in the bag of blood that's being transfused. Those are the most important. Because when worst case scenario, if my red blood cells have a B antigen, and I give them blood that have B antibodies, yes, there will be some clumping, but most of those antibodies will be diluted out. So this is possible. It's not the best scenario, but it is possible. So if you're in a 100-car pileup and the hospitals are overrun with trauma, and all they have left is blood type that has antibodies against you, they can go ahead and give that to you, all right, without your suffering a transfusion reaction. They would prefer to give you your own type. So if you're going to have surgery, and you know it, then in 3 months you're going to have surgery, they'll give you the option of going into the hospital and having blood drawn, spun down, and stored. And then if you need blood, they can go get your own blood and give it back to you, kind of a blood doping. But you're not really you've already lost blood, so you're replacing what you lost, okay? Now, I'll talk about cross and type match. Before they do that, they're going to make sure it's the same blood type, even if it's your own blood. And they will match the two. They'll physically mix the blood together to make sure there aren't some smaller incompatibilities that might result in agglutination. So keeping this in mind, that we can never receive blood if we have antibodies against it. So I'm switching those columns. Now let's put receive from up here. So if my antibody is B, I cannot get any blood type which contains B antigens. Does that make sense? All right, so I can certainly receive blood from myself. So I can receive blood from A. I can't receive blood from B. I can't receive blood from AB because I have antibodies against the B antigen. But I can receive from O because O has antibodies against me, but I don't have antibodies against O antigen because there is no antigen. All right? So I'm not looking at the antibodies here. I'm looking at, does this blood type have antibodies against my antigen? So blood type A does not have any antibodies against A. It has only antibodies against B. Blood type A has antibodies against B. That doesn't work. Blood type A has antibodies against B. That doesn't work. Blood type A has antibodies against B. There is no B. That's why O works. Okay? So I can receive from blood type O. Same rules the rest of the way. I can be blood type B has antibodies against A, but I can still receive B. I can't receive AB because I have antibodies against B. All right? I have antibodies against A, so I can't receive AB. But the same reason I can get O. I have antibodies against A, but O doesn't have A. So I can receive B, O. Okay? Now, I have blood type AB. When I jump in that pool of blood, are there any antibodies there at all? No. All right, so I can receive AB. I can receive A and B and O. Even though O that I'm getting has antibodies against me, it's not the ideal situation. It'd be better to get A or B, but those antibodies will be diluted out. Okay? So as long as I make that prerequisite. Now, if I am blood type O, what antibodies do I have? A and B. So I can't receive any blood that has antigen A or antigen B in it, because I have the antibodies. My pool of blood has both antibody A and antibody B. So I can't receive anything that has A or B. Does that make sense? Because if you understand that, you can figure all of this out. So I can only give to myself. You can only give O. All right?
Wow.
And if you forget that for make sense, make a drawing. Say, okay, here's the blood type. Let's say it's blood type AB. So blood type AB has what antibodies? None. AB has no antibodies, so I can receive anything I want. Blood type A has B antibodies, so this can't have B in it. Anything up there that doesn't have the letter B in the blood type is acceptable. Okay? All right, now let's look at who I can donate to. It's the same principle, but in reverse. Now the blood I'm donating to is the one that can't have any antibodies. So now this is me, and I'm looking at who I'm donating to. So let's use the diagram over here for the first couple just to switch our brains. So if my blood type is A, I cannot donate to anybody that has antibody A in it. This would not exist.
So you You just donate to yourself and A.
As long as there's no A, I can have antibody B, or I can have no antibodies. B or AB, exactly right. So they can have antibodies against the other. It can be my blood type. So it can be, I can donate to A, or I can donate to somebody that has no antibodies against A, and the only other blood type is AB. Okay? If my blood type is B, I can donate to B, because they don't have any antibodies against me, or to AB, which won't have any antibodies against me. I will against them, but remember, it's okay. It's getting diluted out. I can donate to,
AB. Oh.
if I'm AB,
Oh.
I can definitely donate to myself, to AB. All right? Can I donate to anybody else?
No. Because the B antigen.
If I donate to A, there's B antibodies against me. If I donate to B, there's AB antibodies. I mean, there's A antibodies against me.
So no, you can't.
If I donate to O, that's the worst case scenario.
You get both.
They have both, all right? So I can only donate to AB. Now what about if I'm O? I don't have any antigens. So nobody's going to have antibodies against me. I'll have all the antibodies, but again,
You can
in
donate.
the worst case scenario, they will get diluted out, all right? So I can donate to myself, to
A,
A,
B, and
to
AB.
B, and to AB. All right, so that's the question that you have to ask yourself about the donation and the receiving. The big pool of blood is in the individual. That cannot have antibodies against the antigen in the blood bag. Whether you're the antigen, whether you're the blood in the blood bag donating to, or whether you're the blood in the body receiving from, the body can't have the antibodies. Okay, no antibodies in the body. That's prohibited. All right, let's add to this now our D antigen. So D antigen, you have to be exposed to develop antibodies against via a blood transfusion. Eating the antigen doesn't work. So you either have to be exposed to it via a blood transfusion, or you have to be exposed if you're a mother and your blood type is negative, and you have a baby whose blood type is positive. At the time of birth, the fetal blood cells, because of the separation at the stratum functionalis and basalis, the fetal blood cells and the tearing sometimes of that tissue can be introduced into the mother's bloodstream. And even though that baby is safely out of the uterus, the mother mounts an immune response and makes antibodies against the D antigen. Those antibodies can cross the placenta. All right? The A and the B are too large. They're a different cluster. They're actually a cluster of 5 antibodies. They cannot cross the placenta, but the D antibodies from the mother can cross the placenta. So her second pregnancy, where the baby has D antigen, the mother's antibodies can clump and agglutinate the baby's red blood cells and result in death before the baby is born, or a very sick or dying baby after the baby is born. This condition is known as erythroblastosis fetalis. This is not fetalis as in a fatal accident, though it can be fatal, but as in fetal. We're thinking about spelling it. If the mother is positive, if she has the D antigen, there will never, ever, ever, ever be a problem. Okay? It doesn't matter what the father is. can be negative or he can be positive. It won't matter. He can have the D antigen or he may not. Because if the baby is positive and his blood cells are mixed with the mother's, it's exactly the same thing. Like giving yourself your own transfusion. So there's no recognition of a foreign molecule after the baby is born, and the mother does not mount an immune response. Okay? If the mother is negative, bless you, and the father is negative, this is a recessive trait. All right, so I don't know if you remember genetics. They can only have a negative baby. If a baby is positive, somebody's been sleeping around. All right, so the baby has to be negative. No problem. Because there's no antigen to recognize as foreign. So again, the mother does not produce an immune response. However, if the mother is negative and the father is positive, either homozygous or heterozygous, and the baby is positive, then there can be an immune response. Okay? Again, if the baby is negative, no immune response. But if the baby is positive, and in either of these, in this condition, all of the babies will be positive. In this condition, only some of the babies will be positive. All right? So if the baby is positive, and when the baby is born, some of those fetal red blood cells will enter into the mother's bloodstream and interact with her macrophages and B lymphocytes, and she will make antibodies. That baby is pretty safe because he's already out of the womb. All right, there's no way the antibodies are going to cross the placenta if he's not there's no placenta anymore. However, the memory cells making these antibodies remain behind. And the second time there is a baby, those placentas, if it's positive, those antibodies will cross the placenta and it can start, to agglutinate its blood cells. So there is an immune response. Antibodies develop. Usually takes approximately 72 hours for this to occur. The cells that make them, the B cells that make them, remain in the mother's body, and the second time there's a baby, the antibodies cross the placenta and start agglutinating those baby cells. And so they can be born stillborn. They can be born in dire need of transfusion. So when these babies started, the whole antibody blood cell thing was first discovered, researched in the early, like 1901, 1902. And Len Steiner was trying to discover why these blood products were helpful to some people and seemed to kill other people. And so they started researching, and they found the A, B, and the O. But then there were still babies that were having problems in the 30s and 40s, so they researched monkeys and some prisoners in Tennessee that time off for donating their blood and being part of research, early parole. And the D factor was called the RH factor because they worked with rhesus monkeys as primates, and found the D antigen. So nowadays, well, in the early 80s, late 70s, when I started working as an RN,
Got a shot?
they were treating newborns. Babies that were born dead, they obviously couldn't do anything about. But if a baby was born suffering from erythroblastoma fetalis, they would give it a transfusion. So let's say that the mother's blood type is A negative, and the baby's blood type is A positive. What blood type would they give the baby? A negative or A positive? They would use A negative. Even though the baby's blood type is A positive, it's not the A negative of the mother that's the problem, it's her D antibodies that are the problem. So they would give the baby A negative blood from somebody else that had no D antibodies on the chance that there might still be some of the baby's, if you give them positive, there might still be some antibodies left from the mother because they don't completely replace the baby's blood when they do this transfusion. And the blood that's still in the baby may still have some of the mother's antibodies and thus agglutinate the new transfusion. It'll probably get diluted out, but as a practice, they're going to give it negative blood that just has no D antigens and not even run the risk of having any antibodies remaining behind.
Can this ever affect the first pregnancy?
Yes or no? I'll give you an illustration, okay? So they started out doing the transfusion, and then they went to giving a transfusion before birth. So before the baby was even born, they would send a long cannula through the abdominal wall, the uterus, the amniotic sac into the baby's abdomen, and give a transfusion before birth. They were still losing babies. So material that was developed, identified as ROGAM.
I got that shot.
That stands for gamma globulins, which are antibodies. So ROGAM are antibodies against that match the D antigen. And this was given to the mother that was O negative or B negative or A negative gave birth to a positive baby. They would test the baby's blood type as soon as it was born, and they would give this ROGAM shot within 72 hours. The purpose is to destroy, agglutinate any of the baby's red blood cells in the mother's circulation before she had time to recognize that they were foreign. So the next time she got pregnant with a positive baby, it would be like it was her first. She wouldn't have any cells that had developed antibodies against it. Does that make sense? Now they give that injection. They don't wait for the baby to be born and be
There's
blood
two.
type. They give it twice throughout the pregnancy, just in case.
Yeah, my first child, or my first one, she was, so I only had to have the one shot, but then with my last one, I got it midway through, and then when she came out,
And then when she was born.
and then I had to have another shot when she was born.
And that suppresses your response. So you should not have the antibodies, all right? Because if you give it within that first 3 days, it takes that long for the mother to mount the primary immune response, and it destroys the baby cells in her system, so she never has that opportunity. Okay? The question was, can you ever have the immune response with the first baby? The key here is, depends on if you've had a previous exposure. So there was a case where a woman had a baby. The baby was negative. So the baby had no problem. She had a second pregnancy, and the baby, when that baby was born, the baby was positive and had erythroblastosis fetalis. And so the doctors were like, is this your your other baby's negative? And she said, yes. I said, have you had any abortions? And she said, no. No other babies? No. And they couldn't figure out how this first pregnancy that was of a positive D antigen could have erythroblastosis fetalis. And what they finally, I don't know if they ever proved it, but what they finally decided was her first pregnancy must have been a twin pregnancy. And the baby, the other twin was a fraternal twin and had the D antigen but died in utero, few weeks, still had blood cells, and those were absorbed and an immune response was mounted against it. It could have been that, or it could have been just the woman was pregnant, didn't know it, and the baby died in utero, say 5-6 weeks, and she never even knew she was pregnant and just didn't know to report it. Because it wasn't really a miscarriage, it might have just been absorbed, or she didn't realize that it was a miscarriage. So, yes and no. It might appear to be the first pregnancy, but there was a previous exposure, just an unborn child that resulted from it.
Would it have to be pregnancy? You could get it if you just had a blood
You can get a transfusion, but I'm assuming
Using a needle or
there was
something
some
and
for
then-
exposure, yeah. So certainly, a guy can develop antibodies against the D antigen from some exposure to blood or blood product that has the D antigen.
I have watched, when I'm big on the science